Hey everyone, Connor Wander here with the Straight From a Scientist podcast. This is yet another episode from the San Diego trip I took. This one was recorded at Society for Neuroscience conference again. We're going to have one more episode past this that was initiated at the conference, and then I recorded over Skype, so I'm super excited to release that. Again, I'm sorry these are taking so long. School is always going to come first, and the deadlines just stack up. That's how it goes. We'll talk more about that later, but this episode is about GWAS. Now, GWAS stands for Genome-wide Association Studies. Essentially what those are these huge genetic screens. So we've sequenced the human genome, and we're starting to pick out which genes or gene clusters correlate with disorders or diseases. In this case, Dr. Liz Tunbridge studies disorders and diseases of the brain. So she's a psychiatry researcher, and she uses GWAS to help understand psychiatry research. And the reason I talked to her was because I was on Twitter during the conference. A lot of great things were happening on Twitter. People were highlighting really promising talks and really great research posters that you should check out on the floor. There's so much happening that it's impossible just to peruse the program and find out everything that you should be doing. So I was watching that, and Dr. Tunbridge had this really great string of tweets discussing the limitations of GWAS, the promises of them, and just kind of a very nuanced discussion of GWAS. So I reached out to her, and luckily for us, she agreed to come on. So we met in kind of a random spot in the conference areas. I didn't have a designated recording room or anything, so I really apologize for the quality. You'll notice it at the beginning, but it's mostly edited out for the rest of it. And Dr. Tunbridge was just a great sport through all the interruptions we had. Again, I edited those gaps out, but people were just rolling trash cans by all the time as soon as we started the recording, even though it had been pretty dead silent right before that. So apologies, Dr. Tunbridge, again, and thank you for being such a good sport about this if you listen to it. Anyways, it's a great podcast. It gets into the nuance of genome-wide association studies, again, like those genetic linkers that we might one day use for medicines or for diagnostics. So those are the two main applications for these sort of genome-wide association studies. They can help us identify things we should look into further, right? Or they can just be markers. They can just be tags of an increased risk for one disorder or the next. But there are some real limitations, and while they are very helpful, GWAS are not the end-all be-all. And that's what we get into in this podcast. I want to make one more quick announcement. We have officially launched the Straight From a Scientist network, the SFS network. You can find that at straightfromascientist.com/SFS network. This is basically just a grouping of scientists, blogs, other podcasts, and just general tools and resources for people who are scientists or science interested, wanting to learn more about the field. Maybe you're a student trying to find that perfect fit for a research interest. You don't even know where to start. So head on over to the SFS network. We have a great grouping of scientists who we've worked with or plan to work with in the future. And if you're interested in joining, please email us at straightfromascientist@gmail.com with a bio picture, as you see in the thumbnails, something like that, and a little preview for whatever you're working on. And of course, any social media links or places you want to forward people. And with that, give it over to the episode. Enjoy, everyone. Welcome, everyone. I'm at Society for Neuroscience. I'm actually overlooking the main poster area where you can see thousands and thousands of people walking past. So when I snap a picture of that, you guys can understand where we're at. But I have with me Dr. Liz Tunbridge from Oxford University, who caught my eye actually on Twitter with a Twitter rant talking about GWAS in science and all of the caveats and all of the things we have to consider moving forward when we start to use GWAS for science. So GWAS stands for Genome-wide Association Study. And people often say GWAS study, which is kind of like saying ATM machine, but GWASs are really important for us to find genetic targets for possible disease therapies. But there's, of course, some problems with that. So, Dr. Tunbridge, thank you so much for taking the time to come talk to us.
Oh, you're welcome.
What do you think about when you start looking at GWAS studies? Like, what got you to do that Twitter rant in the first place?
So I should start by saying that I think GWAS are a very good thing, and they've definitely transformed my field. So I guess, so maybe I should start by giving you a bit of a background to psychiatry, because it's a slightly unusual field. So in psychiatry, we've known for a long time that genetics are important in determining who develops these illnesses. They're not the entire story, but they play quite a significant role. But what we haven't known is the specifics of that, so which bits of the genome are important, which genes are important. And so what GWAS and other large-scale genomic studies have done is to provide us with that information. So in essence, what they do is they take a really large group of patients and a really large group of controls, and we're talking tens of thousands of people. These are huge studies. And they compare statistically across the entire genome, looking at the frequency of what's called SNPs, or single nucleotide polymorphisms. Those are just single letter changes in the genome sequence, essentially. And they compare which forms are present in the controls and which are present in the patients, and they find associations between individual letters in the genome and illnesses, essentially. So they're fantastic for someone in my field, and I think for me it's a hugely exciting time to be working in psychiatry, because we're kind of moving from an understanding that genes are important to understanding, which genes, and that's hugely important, because we still know an amazingly small amount about the biology of these illnesses. And so the genetic studies have the potentials to kind of give us an unbiased way to look at the biology. However, as you kind of intimated at the beginning, I think these studies are hugely important, but they're not the be-all and end-all. I think the real challenge for us is to take this information and turn it into something tangible, insights into the neurobiology of these illnesses, and ultimately better therapies, which we desperately need in psychiatry. So I guess that's sort of where I'm coming from. My perspective is very much, how do I take information from these studies and use it to understand illness and improve treatments? So I guess that's sort of where I was coming from with that.
No, I thought they were great thoughts, and something that people, researchers like myself, where I don't perform GWAS experiments at all, but I do kind of identify possible targets from them, and I do get ideas from them. So it's good for me to kind of look at that and say, maybe I shouldn't just run away with it as soon as I see a hit, which is what someone's calls like a link, association link in a GWAS screen, versus like the disease phenotype, which is someone having a mental illness. So what types of mental illnesses are you looking at in your research?
So my research focuses primarily on bipolar disorder and schizophrenia, but actually the genes that I'm working on have fairly broad associations with a range of psychiatric illnesses. And so one of the attractions of working on the genes that I'm working on is that potentially, if you can target them therapeutically, they could have fairly broad therapeutic impacts, essentially. So I guess that the whole idea that a family of genes is associated with not one illness but multiple illnesses kind of gets to the heart of some of the challenges we have in psychiatry, which is that there's huge overlap between the illnesses. So many symptoms are shared between psychiatric illnesses, and although at the moment we have these diagnostic criteria and diagnoses that are reliable, if you ask multiple clinicians to diagnose somebody, they will, broadly speaking, come up with the same diagnosis, but we don't know if it's in any way biologically meaningful. And actually one of the things that genomic studies like GWAS have the potential to do is to help us try and break down these complex phenotypes into something more biologically meaningful. But that's still very much in its infancy at the moment.
Right. And so I don't think it's happened yet, but do you envision a future in which GWAS is in some ways aiding diagnosis, or do you worry about confounding?
I think that's a really, really interesting question, and I know it's one that's quite controversial in the field. So there are some people that think that having this information will help us with diagnosis. I suppose I'm fairly agnostic about that at the moment. I'm going to sit on the fence with that one. The reason for that is that because there's huge overlap between diagnostic criteria, and because there's massive overlap between the genetics, in order for that information to be useful, it needs to predict who's going to respond to treatment in some sense or another, because otherwise you're genotyping people for no particular reason, really. And I think we're still quite a long way away from that being in any way a meaningful thing to be doing in the clinic. But we shall see.
Yeah, I asked that because I know a lot of people are going to think about like 23andMe genetic tests and all of these screens where you can actually genotype yourself now and then look up these risk factors. I don't want anyone like doing that, seeing they have a polymorphism, and then completely running away with it.
I think certainly what I can't see happening is you being able to genotype anyone for a single nucleotide polymorphism and conclude anything meaningful about their liability to any of the psychiatric illnesses. We know pretty certainly that no common polymorphisms have a substantial impact on risk for psychiatry. They would just get selected out of the population quickly. So I think we're a really long way away from being able to look at people's genetics and predict their risk for developing any of the illnesses. And one thing we do know from the large-scale studies is that you're never going to be able to predict that from a single nucleotide polymorphism, one of these base changes individually, because they have a tiny effect on risk. The GWAS studies show that quite convincingly, that the common polymorphisms, the odds ratios are typically sort of 1.1 at most. The odd one might be 1.2, but they have a really tiny effect on an individual's risk for developing illness.
So what are some of the more major factors in psychiatric illness? What are the stronger reads on someone's risk?
So what we think is the case is that actually it's not any individual polymorphism by themselves, but actually the sum total of what you have. So if you take the top 10% and the bottom 10%, so people derive these things called risk profile scores, polygenic risk scores, and these are essentially kind of adding up all of the individual polymorphisms, and it's slightly more complicated than that, but essentially counting how many risk alleles an individual person has. And that's essentially a normal distribution. There'll be somebody in the top 10% and somebody in the bottom 10%. And the odds ratios between those groups are much bigger, like 12 to 15, for example. So we know that the majority of an individual's risk of developing particularly schizophrenia and bipolar disorder, these are really heritable illnesses. So the heritability estimates are around 70 to 80%. So it's a really major factor. And the majority of that risk does come from these individual polymorphisms, but they each have a tiny effect. So it's kind of a cumulative thing. How many have you got together? And it's also possible that, and in fact probable, I think, that some of them will have bigger effects depending on some other factor, environmental or genetic, that you may be exposed to. There are also rare variants that have a much larger effect on risk of developing an illness, but as their name suggests, they're rare. You're kind of one in thousands and thousands of people. So in most people, those variants are much less relevant. So we think that the majority of the genetic burden really does come from these common SNPs. It's just, how many you have overall.
Yeah, that reminds me of my field, which is Alzheimer's disease, where GWAS is commonly used to identify major proteins that are different in one population versus another. ApoE4 is making headlines recently as the most impactful risk variant. So if you have one version of ApoE4 versus the normal ApoE3, ApoE4 confers, I think, a 4 times higher risk, whereas if you had 2, it's compounded to 10 times. If you have ApoE4 plus like a TREM2 variant, it's an even higher risk. And so TREM2 being another hit on the screen. So these compound, but none of them alone, or even if you had ApoE4 and TREM2, would be 100% guarantee that you're getting Alzheimer's.
Exactly. It's a similar story in psychiatry, except that we don't have an ApoE4. There's nothing that has that big an effect on risk. So, but I think that probably the general principle is going to hold true. I guess one thing that's important to note at this point is that I've been talking about the outcome of GWAS as identifying genes for schizophrenia and bipolar disorder and so on, but actually that's not really accurate. What they do is they say this particular region of the genome is statistically associated with disease, and we don't know whether the polymorphism in question is is functionally meaningful, if that's actually the causative polymorphism or if it's just tagging something.
Right.
So a real challenge in psychiatry generally is that the SNPs that are coming out of these studies, the vast majority of them are non-coding. So they're not directly affecting the activity of the protein or any protein. And so we think what they're doing is regulating aspects of gene expression. And we tend to assume that they're regulating nearby genes, but that remains to be proven. So although we talk about GWAS as finding genes for schizophrenia, that's not really what they do. They just say this bit of the genome is important, and then we have to work out why. And that's, I guess, that's really what my research aims to do, at least the kind of first step in that pathway.
Fascinating. So, non-coding regions are another kind of black box in biology that is starting to be opened, and we're starting to figure out what they do, but we aren't exactly sure, and the more we learn about them, the more we learn that they are having an impact. Right? So
and it's all incredibly complicated, yes. That is definitely true. I've been wandering around the poster sessions this afternoon, and there's some beautiful work here, kind of looking at new ways of finding enhancers, for example, the elements that are controlling the way in which genes work. But again, that's an area we really are in our infancy of understanding, how you solve that challenge of turning this statistical association, which is what GWAS spit out, into some kind of meaningful understanding in biology.
Yeah, and I mean, this all seems really important in the context of mental illness. Some of these diseases are incredibly debilitating. People can't hold jobs. They can't really socialize properly. But I saw you had a TED Talk, and that's on your Twitter page. It's pinned up to the top. By the way, at Liz Tunbridge. I'll link it in the show notes, and I'll also link this TED Talk. The title of the TED Talk was something like, would there be mental illness in a utopia, like a perfect world? Why do you ask that question?
Well, I asked that question because in my work, I like particularly the field I'm in. I think it's really important to kind of discuss my research with the public, and particularly with individuals affected by these illnesses in some way, either as people who have a diagnosis or family members and so on. And I did a Café Scientifique. I'm not sure if you're familiar with that. It's just essentially a Q&A session. Okay. So I gave a 10-minute introduction to what I do, and then people ask questions. And that was one of the questions I was asked in the Café Scientifique. And I think I did a fairly horrible job of answering it at the time. So my TED Talk was my attempt to make amends for that. So I've become, the other thing about mental illnesses is that the illnesses are what make us humans and make us unique individuals, particularly, so they're disorders of mood, of personality, of, really core human traits. And I've been very interested in the idea, people always ask me, are we going to be able to fix people's genes once we understand exactly how the human genome works, if we ever get to that stage? Will we ever want to kind of mutate people so that they don't develop these illnesses? And my strong feeling is that, firstly, I don't think we're going to get to the stage where we can predict with enough certainty who is going to become ill from their genetics for that to be feasible. But even if we could, I don't think we'd want to, because I think the genetic variants that make us more or less susceptible to these illnesses are highly likely to be the same ones that make us unique individuals. And so, my TED Talk was really an attempt to answer the question I was asked, and answered really quite horribly in that Café Scientifique. I won't spoil what I think. You can go and watch the TED Talk and find out.
People can click the link, absolutely. Well, I've heard that a lot of artists, musicians, painters, poets, it's not poems, have some at least increased risk for some of these diseases. I could be wrong on that. I don't know.
Yeah, I'm not sure, actually. I don't know. I mean, it's anecdotally, there's, particularly manic depression is something that, historically has been linked with creativity. I'm not sure it's been systematically studied. It would be a difficult thing to do, I guess, because you'd have to define artists and non-artists. Right. And I could definitely say I'm a non-artist, but for most people, there's probably a continuum.
Yeah, exactly. That is all subjective too, so there's an issue with that. And then you have an issue with which I see in a lot of other studies that claim, for example, that gamers have increased white matter or whatever. Generally, when you're looking at those types of people, you're looking at high performers, and they're always going to be different because they're outperforming the general population. Really interesting work, and definitely going to watch that TED Talk just because it's been only hours since I reached out to you, and at this conference there's no time for YouTube, but I recommend everyone check it out. I know I'm going to check it out in just a second. Thank you so much for coming on, Dr.
You're welcome.
Tunbridge.
It's been a pleasure.
Before we break, is there anything you would like to pass on to the audience?
I guess I'm assuming that many of your listeners are students who are trying to figure out what they want to do, and
Not everyone, but
people
a lot of them,
who are kind of interested in research and don't know how to get involved. And I guess my advice would be not to worry if you don't know what you want to do, because I've never known what I want to do. I kind of stumbled into research. I'm from a completely non-academic background. My family didn't have a strong history of going to university, and so I have kind of stumbled into research, and I think scientifically I continue to kind of stumble. Although my background's molecular biology, I really like collaborating, and the questions that I'm interested in answering require quite a broad technical approach. And so now I'm really fortunate in that I'm somewhere where I'm able to do that. I can make collaborations with people, and I can use whichever technical approach is the best to answer the questions that I want to answer. So I guess my advice would be, don't worry if you don't know what you want to do, and don't be afraid of not knowing what you're talking about, because I think the most exciting research comes at the interface where two people from different fields are talking and neither is completely 100% sure what they're talking about, but there's something very creative in that gap between disciplines. So I guess that's my top tip. Don't worry too much.
I appreciate that. I was in a similar spot, and that's kind of, trying to help people out with this podcast. And then, to add on to that, kind of asking the right questions thing, the most exciting moments in research are when you and whoever you're working with sit down and look at your data and say, well, we know something is happening, we just don't know what yet.
Yeah. I think the most exciting findings I've had in my research have been the ones that have been unexpected, so actually the data that I've been presenting here were meant to be a pilot study, and actually what we found in that pilot study was so exciting that it sort of snowballed, and it's really shaped the kind of research that I'm doing now. And I think, it's those moments that make all those periods of, PCR's not working and late nights in the lab kind of worthwhile.
Yeah, long hours in the lab worth it. Thanks again, Dr. Tunbridge. Been a pleasure. Thanks for listening, all.
That's going to be it here for us at the Straight From Scientist podcast. Thank you so much for listening, and especially if you provided feedback or are a sponsor of the podcast. If you'd like to become a supporter, you can go to patreon.com/SFS podcast. Another way you can support the podcast is through our collaboration with Plant Warrior. So Plant Warrior produces 100% all natural vegan protein, which contains all 21 essential amino acids, but does not contain any artificial sweeteners like sucralose that have been shown to do damage to your gut microbiome. They also plant one tree for every protein bag purchased. These guys have incredible values. They align perfectly with the Straight From a Scientist podcast, and we would not have them on as a sponsor otherwise. If you want to check them out, you can go to plantwarrior.co/discount/SFS10. Or you can just use the SFS10 discount on their website to get 10% off your order. Again, we really appreciate y'all listening. Hope you enjoy each and every episode.